Alright guys, so today we're going to be doing the mitosis review, which is this mitosis worksheet here. So I have it in Kami and I also have it as a PDF in the assignment folder. So you'll have to email me the PDF or your answers because you won't be able to actually write on the PDF if you click on that one. But you can just type out your answers, what they would be if Kami isn't working for you. So you have two potential ways to do it. If Kami works, great, do it in Kami. If it doesn't work, then just open up that PDF, have a separate Google Doc that you can write your answers down in, and just make sure you number everything and we'll be good. So label the following diagram with the phase of mitosis, A through E, or the type of cell structure, F through H, seen during mitosis. So I'm going to go to a text box here. And for A, so this is starting off at the very beginning, and that is going to be interphase. Interphase. And then right after interphase, we have prophase. Remember, metaphase is where things meet in the middle. And then if you just follow the arrow down, we have anaphase. And the last phase is telophase. As we're looking at these structures here, I labeled centrioles for you already because that one is a little bit difficult to decipher. This one here, we are checking out the middle portion of these chromosomes here. That's a centromere. The next one down here, we are looking at the lines that are coming up and joining up with those chromosomes. Spindle fibers. And the last structure here that we have to look at, it's kind of difficult, but it's pointing to part of the chromosome, and after they're separated, they're called chromatids. What's a chromatid? During which stage of a cell cycle do the replicated chromosomes thicken and become visible? Well, that would be down in prophase. During which stage do they line up at the equator? So when are they lining up in the middle? That's in metaphase. During which stage of the cell cycle do they replicate? That's an interphase. Remember the S phase specifically of interphase. The drawing below has been made of a photograph showing a cell undergoing mitosis. Based on this, which stage would it have been in? Well, I can see that the chromosomes are being pulled apart, so I'm going to go with the anaphase. The drawings A to E show stages of mitosis in a plant cell. So we're going to be labeling. Obviously, they're not in order. So interphase, we've got to find the very first one. Or if you can find prophase first or metaphase, whichever one you... So if we go back up here to our first diagram, this would be interphase, and here is prophase. So if we're looking up here, one of these ones is going to be interphase, one of these is going to be prophase. Let's see if we can figure out metaphase. They're lining up in the middle. So I'm going to go with E for metaphase. And where they're separating, that one's pretty easy. You can see in A, they are separating from one another. Telophase and cytokinesis. That's going to be D. And this one is also going to be D. Back up here, I'm stuck between B and C. The only difference really is that you can see this little dark circle here and these ones, there is none. So I'm going to take it that this here is going to be a centriole that is starting to form, and you can see that the cells themselves are darkening, not the cells, the DNA strands are darkening. So I'm going to go with B for interphase, and I'm going to go with C for prophase. We have the two processes which occur during interphase and are necessary for mitosis to take place. The two processes, well, we know that it has to grow, the growth, and blank of the DNA, replication. This drawing shows various stages of mitosis in a fast-growing onion root tip. Identify the cells by number. They're in the following stages. All right, so prophase, metaphase, interphase, telophase. Well, I always like to start with metaphase because honestly, it's just the easiest one to see. So anytime you see a cell lining up in the middle, that's going to be cell 1. Nothing in that second row. 11 and 17. Anaphase is also super easy to tell because they're being pulled apart. So we got 7 and 9. I didn't realize that my cursor was on the wrong spot. 9 and 7, same deal. And telophase, that's where we have new cells forming. So we got 3 and 12. Okay. So interphase is where you can't really see what's going on. Prophase is where you can start to see those individual chromosomes. So I can't really see what's going on in number 4. All I see is the nucleus. The nucleolus has not broken down yet. So 4 is going to be interphase, along with 5, 6, 8, 10, 13, 15, 16, and 18. And the majority of your cells, if you were to look at any given specimen, you would find that the majority of them are actually in interphase. Prophase, we got cell 2 and 14. I think I got them all accounted for now. Let's see, I could count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. All right, we're good. Using colored pens or pencils, show how 2 chromosomes are passed from one parent cell to the 2 daughter cells. I have to take down that a little bit. Okay, so 2 chromosomes pass from parent cell to the daughter cell. Why isn't it drawing? Maybe I have white. Maybe that's the problem. Let's move that up. Yep, I'm on white. Okay. So here I have a chromosome. Here I have a chromosome. And now I'm going to put my spindle fibers in there. I've still got my chromosomes. Got 2 in there. This one's going to get 1. This one's going to get 1. And eventually they'll be divided. All right. That looks pretty good to me. If you guys have any other questions, feel free to shoot me an email. Again, if it's not working for you, please use the PDF and just get a separate Google Doc, number your answers, and send me an email that way so you can still get credit. Don't get focused on the fact if Cami isn't working for you. Please don't spend hours trying to make it work. Just Go through, get the assignment done the easiest way you can. All right, guys, I'll talk to you soon.